Welcome to Cut the Crop, keeping you in the know about everything you grow. Researchers, growers and other interesting folk delivering insights and information on topics ranging from agronomy to profitability. Brought to you by the Foundation for Arable Research.
Hello and welcome to Cut the Crop. I'm Anna Heslop and today my guest is Penny Tricker. Penny is a science group leader for the Cropping Systems and Environment Group at Plant and Food Research. Welcome.
Thank you.
Now, Penny, you've not been at Plant and Food for terribly long. Can you tell us a little bit about your background and how it led to your arrival in New Zealand?
Yes, so I have been at Plant and Food just over 3 months now. I came from Australia, where I was working at the University of Adelaide in South Australia for 11 years. Before that, I'm originally from the UK, as you can probably tell from my accent, and studied at the University of Southampton and University of Reading. And my work has always been on the effect of environment on plants. So in particular, in Australia, I was working on wheat, barley, and grapevine. And for wheat, looking at how we could produce good quality and as much wheat in very dry and hot environments like we have in South Australia.
And some of that was also around potential impacts of climate change.
Yes, so again, throughout my career, I've been looking at the impacts of climate change, whether things like the increased amount of CO2 in the atmosphere or water stress or what the next limiting resource might be. So, for example, soil nutrition, but particularly always with a focus on water stress.
So what do we know about those potential impacts?
Well, we know that it's going to get a lot more variable. I think we're already seeing that. So the predictions for 2050, best case scenario, if we all limit our emissions, as we should, under the Paris Agreement, we'll still have 15 more days of extreme heat in New Zealand. We'll still have more floods, more droughts, and that whole cycle of water being very different and unpredictable. And we will have more events like fires, unfortunately. So it's more of all those bad things, basically.
And they are obviously going to impact on crop production.
Yes, they are going to impact on crop production. Looking at options for that, of course, you can say, well, we move with climate change. So perhaps we move some of our production to different areas. Perhaps we change crops. But overall, the impact of particularly water stress is always a problem for any crop.
So how can research help?
Well, so several ways. One of the good ways to do that, particularly with cereal crops, is to feed into plant breeding. So if we can find, for example, a wheat that is a line from a dry and hot place that does pretty well, and bring that into a breeding program for wheat which does well in New Zealand and cross the two, hopefully through plant breeding, we can still maintain some of the yields.
And so how much of that plant breeding work and investigation is already underway and how much is to come?
Oh, that's a good question. The answer is a lot is underway. So if we're talking about wheat around the world, it's a big focus of research, particularly to try and combine drought and heat tolerance, because, of course, hot and dry weather tend to come together. So although we have many years of research on, for instance, drought-tolerant wheat or heat-tolerant wheat, putting the two things together and still maintaining yield and quality is quite a difficult challenge. So there's a lot going on around the world, particularly at the CIMMYT Center in Mexico, but also in places across the North American plains now where they've had terrible drought and heat stress the last couple of years. In Europe, of course, big producing wheat areas like France, where again they've had a lot of drought and heat stress. And for sub-Saharan Africa, there's always attention there. New Zealand, I think, has been a bit sheltered from that so far, in that although it is getting hotter and drier, and particularly nights are getting warmer overall, hasn't had a big effect on yields yet.
So will that international research, is it relevant, directly transferable to New Zealand?
That's a good question. And I think the answer is some of it, yes. So the fundamentals of research, if we can understand, for example, what makes a plant more drought tolerant, then that can be transferred all around the world. But we do still want things that do well in New Zealand. So it's bringing that international expertise into New Zealand, but also listening to the researchers and the growers in New Zealand about the specific circumstances here and combining those two.
So on that note, how relevant is the work that you were doing at the Wheat Hub in Australia? What lessons can you bring from there?
Well, I think very relevant, because what we were doing there was developing the lines in the first place. And so what you can do is take, as we did, hundreds of different wheat lines from around the world, make all the crosses and associate the differences between what you see with those lines with the genetics. And when you can bring those two together, then it's transferable. So that's one of the ways to do it. The other things we were developing there, apart from the genetic information and those tools, was phenotyping. So by phenotyping, I mean measuring traits in the field. And this is a huge challenge at the moment, because if you're looking for hundreds of differences in thousands of lines across hundreds of thousands of hectares in Australia, you need high throughput ways to do that. And so we developed all the algorithms where you can capture a picture from a drone and use an algorithm to determine the measurement. That's transferable anywhere.
That's really high tech.
Yes, very high tech. And we had some fantastic new instruments and machinery coming out of that. So where you can scan across a field with hyperspectral cameras, or we used X-ray computed tomography to actually look at seeds developing inside by X-ray in wheat heads. So these kinds of things are very high tech, and I think agriculture generally is becoming more high tech. The good thing about Australia is because the growers there have huge challenges, they really are farming in very, very difficult conditions. They're early adopters. And I think that's something that it's quite useful for New Zealand, that you can try things out in Australia and then bring them to New Zealand.
So were there any specific crops that you were looking at in Australia that gave you some ideas going forward?
So my sort of hobby, I suppose, crop was durum wheat, so the wheat for pasta. And what was really great there was to be able to use the wild relatives of wheat, so grasses basically that are collected from all around the world, and look for these beneficial traits in those wild relatives and then bring them into modern durum wheat. That's a very long work, again, internationally with lots of great collaborators. But we also use that with some researchers from New Zealand, where they had found that beneficial fungi that help plants to establish would only establish in wheat when it was a wild relative, not when it was the cultivated variety. So we were working with researchers in New Zealand to bring that into modern wheat.
That's part of the Endophyte project, which
Yes.
I was involved with
Yes.
as well.
Yes.
That's a chat for another day.
Yeah, no, it's very cool research.
And was it just wheat, or what about barley? That's the other major cereal crop here.
Yes, yeah. So I worked on barley quite a lot as well. Not on the endophyte work, actually. That was specific to wheat. But barley, we were working on something called epigenetics. So I don't know if you know what that is.
I think it's a good idea if you explain it.
Yeah, so what we mean by epigenetics, epi means above in Greek. And so you have genetics, you have a gene, and that's kind of like a light bulb in your house. But to switch it on and off, there are various modifications that are actually chemical modifications of the DNA that can turn that on or off or up or down. And that's what we mean by the epigenetic profile. And so there are little bits of RNA, which is the bit that comes from DNA to code for a protein, that will attach to some DNA and make it more closed. So you won't get that gene being what we call expressed, so it won't get turned on. So that's what I was working on in Bali. And that's really powerful. What we know now that perhaps we didn't know 20 years ago is that genetics isn't everything. It's about how it gets switched on and off and up and down. That's also important, especially for yield.
And what about, you mentioned earlier, one of your major interests, and obviously for the world as climate change progresses, whichever way it goes, water and water use efficiency for crops.
So I currently still work in water use efficiency for wheat, but I have worked on water use efficiency or efficient use of water. They're sort of technically slightly different things in science in a lot of crops. And this is vitally important because with climate change, fresh water is going to become a limiting resource, and agriculture is one of the biggest users of fresh water worldwide, including in New Zealand. So there are things that you can do about using water well that perhaps we should be thinking about now for climate change, whether that's making sure your irrigation is as good as it can be, whether it's considering how you're going to store water when you have these cycles of lots of rain and then nothing, and also looking at a crop and saying, how can we change its water use perhaps so that it uses water loses water at a cooler time of day, like at night, for example, which will help with heat stress, these kinds of things. So one of the ways we've been doing that is by looking at those modifications in DNA that help. What we were doing with wheat was taking a bunch of seeds, mutagenizing them. So basically that just means you stick them in a harsh chemical that's going to make lots of little changes to their DNA. Wash that off, grow them out, and then you look for how is that one doing compared with another one, and say that's to do with that particular mutation in the DNA. So then again, we know that mutation, we can potentially use that change going forward.
So that leads on to another very, very heavy question around when does that research become genetic modification and when does it not? And can all of this be done without genetic modification? How do we sit on that?
What I would say there, that kind of mutagenesis that I was talking about is happening all the time. In a wheat field, you would find more natural mutations as the seeds are formed than we would find from doing it that way by chemical mutagenesis. So, and it's been done for 60 or so years, at least, around the world, this kind of chemical mutation or irradiation mutation, bringing in from one species to another. So, for instance, most of the bread wheat that we currently use around the world has a bit of rye in it. So it's controversial, but not really. What's controversial is the method used. And so the method of genetic modification that has been around now for 20, 30 or so years was what they call transgenesis, where you're getting something from one species and deliberately putting it into another one. That's actually really, really hard to do, really long, really expensive, quite apart from whatever you think of that method. Now we have the advent of a thing called gene editing. So gene editing actually uses a bit of that epigenetics I was talking about. It uses little bits of RNA to modify some DNA, basically by a natural process. The difference is that we're targeting it. So we know how to target that now. So if you know enough about a gene, you know enough about its code, and you use that method, you can just knock out one or two little bits of DNA and make a big difference. And I think that gene editing technology is probably the way that we're going to go. It's different in different countries, how that's regulated. Of course, as a scientist, I want to see the research continue because that will help us anyway. So, for example, if we deliberately edit a plant in research and we see that has an effect and makes the plant more water use efficient, then we know the code. We can go and find that naturally in another plant.
And that can speed up the natural breeding process.
That can speed up the natural breeding process, yes. The breeding process is long, and genetic modification actually isn't that much quicker, but these kind of gene editing or deliberate mutagenesis techniques are both quicker and they're more efficient. But for the moment, the molecular marker, which was knowing about that DNA and just searching for that in a plant, is used around the world, not so much because it's quicker, but because it's better. It works.
But we should just be clear that that's still not a generally available breeding tool in New Zealand.
Molecular marker assisted breeding, where you're just looking for the DNA, is available in New Zealand and used, but genetic modification, including gene editing, is not at the moment. It's only used in research.
So here's the big question. What do you think a wheat crop in New Zealand might look like in 20 years?
I think the yield will be a little bit less, I'm afraid. New Zealand has record-breaking yields compared with most of the world. So I think whatever we do, we probably have to accept that yield per hectare is going to be a bit less. But I still think it will look good. And I think the quality will have improved because of the advances in breeding.
Well, we'll end on that really good note. Thank you very much, Penny. And listeners, have a great day on the farm.
Thanks for tuning in to Cut the Crop, presented to you by FAR. If you'd like to know more about any of the topics discussed or have any suggestions for future topics, go to www.cutthecrop.co.nz.